All right. Yeah. So before we get started, I would like to just do a short, very short advertisement. I put it into the chat box about a symposium that's coming up on April the 21st. This year, we have this symposium every year for 7 years. This year, our theme is AI in medicine and healthcare. There's a free registration. It's an in-person event on UAB campus. I think feel free to browse the program, the preliminary program. Sorry about the dog in the background. And then we're still having an open call for abstract until the end of this month. So if you have something related to this topic, so go online and submit your abstract. Now, I think it's very exciting for me to introduce Professor Avi Mayan. I know Professor Avi Mayan when I visited Mount Sinai about I think 7 or 8 years ago, but I've known of his work for more than a decade. Dr. Mayan is a Mount Sinai endowed professor in bioinformatics. He's also a professor in pharmacological sciences and the director of Mount Sinai Center for Bioinformatics. He is incredibly productive and innovative, in my opinion. There are a few bioinformaticians that I monitor the work from time to time. And Professor Mayan is certainly one of them. And I remember the first time when I look at his work, and he's mostly working on the links databases, and he was the chief of the data coordination center that actually is funded by the NIH. And then over the years, and his work really evolved. He's incredibly productive in churning out the bioinformatics tool that actually people use. For example, the Enrich-R that was created in his lab less than, I think, 8 years ago, now has more than 6,000 citations. And he has a dozen of tools that cover the areas of multimodal analysis, systems network biology. And I think you may easily classify him as a bioinformatician, but his work also covered on the biology side. He is in the news for discovering genes that's related to Lyme disease and so on. And just look at staring at his lab logo, you can see how incredibly innovative he is in coming up with a very interesting logo. And I also would like to mention to the students that one of my students from 60 years ago was looking for bioinformatics educational resources. And the student pointed out that Dr. Mayan's lecture online was very interesting. And so you may find out and then get initial courses from him. Without further ado, let's welcome and give him a really warm welcome.
Thank you so much, Jake. It's really a pleasure to be here. I really appreciate the introduction. It was like one of the most complimentary introductions that I think I ever received. So it was very nice to hear. I also, wanted to visit you in person. So hopefully we'll be able to do that in the future. And thank you all for your time today. So the title of the talk is Computational Screens to Identify Targets for Cell Removal. But I'm going to give you a little bit more of background of how we got to this point where we're trying to identify those targets. And this is going to be the last part of the talk. So first, I will sort of like start from the beginning of my academic career. I was a software engineer in 2 mid-sized companies, and then decided to get a PhD. And I realized that in biomedical research, when you or enter a PhD program, you get a stipend. So that attracted me to biomedical research at the time. And I wanted to combine computer science and biology. And I chose the laboratory of Ravi Iyengar, who was an expert. He had a wet lab and he was an expert on cell signaling and systems biology at the time. And the idea was that we can start constructing cell signaling networks by reading the literature and extracting interactions between cell signaling components. So after a lot of work, we constructed this network from literature where every edge here is a article that describe a ligand binding to a receptor or some enzyme in the cell signaling network that is phosphorylated by a kinase that transduce the information to those machinery components. And this was published in Science in 2005. So it was a lot of work to manually build this network from literature, and it has a lot of limitations. And also realizing that there is a lot of unknowns, and we really could not really understand how this network actually works. So this is a news and views article that was featuring our article in Science, and it just highlight the various types of dynamical models. So at the time in 2005, people still tried to model biological systems as dynamical models, and without much success. So this article that we had was at the end of very simplified dynamical models where we try to introduce like flows into a topology of a network and see how the topology emerges. And since then, I really stayed even at the higher abstract level of biological data, where we're not even trying to do dynamical modeling. We just tried to fill in the information about genes and their pathways and their properties in a relatively static way. But as you can see, that there's a lot of stuff to do. And this is mainly because we really don't understand cells at the molecular level yet. So after I got this paper in Science, it was really important for my career because I was able to get some offers for having my own lab. And then when we started the lab, I decided to stay at Mount Sinai. So I did my PhD at Mount Sinai, and then I got a counter offer to stay there. I decided to stay, but I did not want to continue to do this manual extraction of interactions from the literature. But what I found is that people have started building those databases of, for example, these are 6 databases of kinase substrate interactions that we integrated. So we wrote some scripts to process all this data and combine it into a tool called Kia, where you can come in and you can do enrichment analysis to prioritize kinases that potentially phosphorylate your set of proteins. Very similarly, we also build this other app called Chia, where we collect from the literature transcription factor targets from ChIP-seq publications, and then build this database of ChIP-seq transcription factor target interactions. And then we can come in with your like differentially expressed genes and identify transcription factors that are enriched for those genes. So soon enough, those are 2 apps that were pretty successful. And then we realized that you can actually do this. You can for a lot of other types of data. So the concept, the general concept is described in this review article that we wrote in 2014. And the idea is that there's so many different types of data that is out there that is not integrated. And by writing some code to convert this data into networks or gene set libraries or bipartite graphs, we can start integrating it at either the gene level or the drug level. So these include, for example, a drug knockdown, like drug or gene knockdown followed by gene expression, this is the type of connectivity map data and the data from links. You can also do this for transcription factors and their targets, like we did for chia. And then you can also look at cell viability data, where you have a bunch of cell lines, and then you try to knock down individual genes in those cell lines, like the CRISPR screens, or just try to do drug screen for cell viability. You can also have those associations of genes with phenotypes, basal expression in tissues and cell lines in normal tissues can also give you sets of genes that are associated with specific cells and tissues. Then you have all those databases of protein-protein interactions, metabolic pathways, and cell signaling pathways, and then also drugs that cause toxicity and adverse event. And these are only part of those types of data that are out there that could be integrated. So we've been very successful, like Jake mentioned, building 2 other apps. One is Enricher that is widely used. It already received almost 60 million queries. And here you come in with your gene set, and then you get enrichment analysis against 211 gene set libraries and almost 500,000 annotated gene sets. And with Harmonisome, it's very similar. It's also a collection of process datasets that people can download. And this is some of the Google Analytics from Harmonisome, showing that in the last 90 days, 77,000 users accessed this site. So what we already were thinking in 2014, that you have all of those biological entities, and then you can connect them. So for example, you can connect gene expression signatures to cancer cell lines that were, those signatures were measured in, and then you can take cancer cell lines and find the tumors that are most similar to them. And then those tumors are coming from tissues, and those tissues, you know, they have like genes that they potentially are mutated in disease. And then everything is basically centered around genes, and genes can interact in pathways through protein-protein interactions. And then you have genes association with phenotypes and diseases. And then those diseases have a lot of relationship with the phenotypes. Like, so for example, mouse phenotypes and the diseases can be connected, and then diseases are similar to side effects, and drug cause side effects. So all those things can be connected. And at a more high level, we have like the genes in the center, and then those other entities like cells and tissues, drugs and small molecules, gene sets and pathways and diseases, phenotype, and adverse events that all can be connected to each other to build those bipartite associations. And then I'll discuss a little bit more like why this is so useful. So this was an initial view that we had, and we really focused on gene proteins and targets. And then more recently, we extended this to also consider patients and subjects, assays and experiments, and also viruses and bacteria and other pathogens. So all of those types of entities can be connected. If you think of examples from your research, you know, drugs can be connected to genes by looking at the either drug targets or the drugs that bind to a protein structure, or the genes that are differentially expressed after drug perturbation. And so every pair of those types of entities can be connected. So you can connect the drugs directly to a pathway or a gene set in some ways. And then there are different assays and experiments that you can use to measure those things. So all of those things are sort of like at a very high level can be connected. And the opportunity there is really to build those knowledge graphs. So this is an example of a prototype that we've been building for Enricher to look at how knowledge can span across gene set libraries. So right now, when you do an enrichment analysis with Enricher, you get independent enrichment for each gene set library. but a lot of the times genes that are enriched from your input are showing up across libraries, and there's overlap across those libraries. So this is a way to look at the data in a sort of like a cross-library enrichment analysis with a knowledge graph, and people have been sort of becoming more interested in a knowledge graph, and specifically we are using Neo4j, which is a knowledge graph database that efficiently store the data in that in network format. So this is a slide that was borrowed from Robert Murphy, who is the chair of the machine learning department at Carnegie Mellon, and he's also interested in systems biology. And he was saying that, we have a lot of data that was already collected. So there is a growth in the amount of data that is collected from cells and tissues, but we are not really processing most of it. So the data that we have collected is this yellow circle. And then the data that we actually understand and analyze is this smaller blue circle. And then the data that we really need in order to fully understand the system is this big background light blue box. So there is really still not enough data, but also there is a lot of data that is not really used. And then there is this opportunity here to do data imputation with machine learning. And so this investment of, this is a plot that shows the investment in studying genes. So it sort of like highlights the same point, that there is a lot of knowledge about some genes and potential targets, but a lot of those genes are completely either understudied or completely unknown, like their function is unknown, but they are measured in a lot of those omics experiments. So in those unbiased genomics, proteomics, transcriptomics, those genes have expression. They are known to play a role, but in the published literature, you will not find any individual study that studied their function. So there is this skewness in the amount of funding, as well as the amount of research that is applied to study individual genes and proteins. And this is this plot was created for the Illuminating the Druggable Genome project that I'm also a part of, where we are trying to use computational experimental methods to illuminate knowledge about kinases, ion channels, and GPCRs, which are three families of the most successful drug targets to date. So this is just to highlight of the fact that we have information about a lot of genes that is not really translated into knowledge. And in order for imputing or start doing this discovery based on guilt by association using ML type of or AI type of methods, what we've been doing is using gene-gene co-expression data. And for that, we built this app called Arches4. And Arches4 is a relatively simple system where we just go to the gene expression omnibus, the place where people deposit all of their gene expression data. And specifically, we are interested in RNA-seq data from human and mouse. And then Alex Lackman from the lab developed this pipeline to automatically uniformly align all the data from the GEO database and build a matrix of data from that resource. So currently we have over 1 million samples uniformly aligned across thousands of studies. So if you imagine it like as a matrix, like a big matrix. You have the genes as the rows, and they have some type of a level of expression across each individual samples, across all of those millions of studies. So the column in this matrix is a million columns, and the genes are somewhere around 40,000 genes that are measured values. So what you can do, you can create a gene-gene co-expression network by looking at how genes are co-expressed. And then you can use that information for making predictions about genes that function based on the known functions for the genes that have been widely studied, or genes that have some phenotype that was identified from phenotypic screens or from knockout mice. And this is unsupervised method that where we just take the a gene set, let's say a gene set from enricher, and we ask what collectively on average are the genes that are highly co-expressed with that set. And then we assign the function for that gene. So for example, if we take the gene Clotho, and we look at predicted phenotypes for this gene. So Clotho is a gene that is known to play a role in aging and also in the kidney. And there are some human phenotypes that have been assigned to it in the human phenotype ontology. And then this is the gene sets that are mostly correlated with Clotho. So we rank the gene sets, the terms that are associated with those gene sets, and then we look at how we do when we are trying to see where the known terms for Clotho exist on that rank. So the most increasing circulating greening level is the gene set that has the highest co-expression correlation with Clotho, and then we rank that term as the #1 term. Recently, we published a paper that improves upon this method, and the way to do it, what we've done is that we partition the matrix of co-expression into clusters. And this is to account for the fact that genes are very context specific. And those clusters could be like correlated or related to cell types or specific conditions where genes are acting together. And then we aggregating those correlations. So we create a correlation, gene-gene correlation co-expression matrix from each cluster. And then we do the prediction afterwards using a machine learning method, supervised machine learning algorithm that takes all of those matrices and churn them into a ranked prediction that is better than the global predictions. We also recently published a paper that does this prediction specifically for long non-coding RNAs. And this is the website for that. It's called LinkHub2. So here, what we've done, we took the gene-gene co-expression correlation matrix from ARCHES-4, and then we annotated the functions for 18,705 human link RNAs and 11,274 mouse link RNAs by looking at co-expression. And the reports that are generated also include additional information like cellular localization, the potential drugs that would increase or decrease the expression of the link RNAs, and then some information that exists about the link RNAs, like their predicted secondary structure and other things that are not directly predicted with gene-gene co-expression data. But the point is that you can do those predictions also for genes with no known information or no publications and annotations, and the long non-coding RNAs are this emerging family of genes that are highly understudied. So the next thing to do, and this is a way to improve again the predictions that we've done for using the ArchES4 resource, you can concatenate features about genes into a machine learning framework where you have the genes as the rows, and then you can have transcriptomics data, like the ArchES4 data, you can include proteomics data, you can include perturbation data, like the links data, and then also literature data, like pathway information. And then you build your favorite machine learning model, and then you can use data about known functions for genes. For example, here is what will happen to a mouse if you knock out a specific gene, and some genes have been knocked out in the mouse, but not all genes and not all conditions. So there is a lot of genes that haven't tested yet. And then what you can do based on this skilled path association, attribute accumulation, or properties from different sources, you can concatenate those as features to make predictions. And this can now be done relatively easily with different libraries like TensorFlow, scikit-learn, and then you can also apply deep learning to this type of exercise. And the other thing that you can also do, which we've done for drugs, you can make the drugs the rows of those models, and then you can concatenate features about the drugs, like their differential expression after drug perturbation, their changes in morphological features. You can include chemical fragments, or you can have like those cell-based assays for thousands of drugs. And then in this particular paper, we tried to predict side effects. So we did this multi-label classifier that predicts side effects, and this works really well for some drugs. So those violin plots show that there is this bimodality of the quality of the prediction, suggesting that for some drugs, this approach works really well in predicting side effects, and for some it doesn't. And this is likely because some drugs work in a systematic way and may not, there is a pro drugs, and there's other caveats of how drugs actually induce their side effects. It could be like a long-term effect that is not detectable based on the features that we tried here. But you can see that for majority of drugs, this actually works. So we can predict the adverse event or the indication for preclinical drugs based on existing drugs. So this is, again, this idea of like imputing knowledge based on existing knowledge. So now I'm going to shift focus a little bit, and I'm going to talk about another sort of like stream of progress that we've made through the years with this idea of making research accessible through changing the way people publish data and perform bioinformatics analyses. So this is the first step for that was a paper that we published in this F1000 research, where we wanted to have the first published paper that is also a Jupyter notebook. So we did the RNA-seq analysis for a recent published study of Zika virus, and Zika at the time was the virus that made people nervous, and it was really affecting countries like in Brazil. It was like a big deal, causing birth defects. So we took some published study, and then we built this Jupyter notebook that shows how you can analyze the data, if all the steps from the alignment to normalization and differential expression, and then enrichment analysis, pathway analysis, and also finding small molecules that can reverse the expression signature. So this paper is a regular paper that has an abstract and figures. but it also a Jupyter notebook where you can download the paper and then you can potentially use your own RNA-seq data and rerun the pipeline. So then a few years later, Dennis Torre came to the lab and he built this app called BioJupies. And BioJupies is sort of like a the next step for this idea of like automatically generating workflows that look like papers. So here you can start with your own RNA-seq data, or you can fetch studies from Geo, or you can just upload your data matrix. And then similar to the way you fill out your taxes, you go through a bunch of questions about your data. You have to put some metadata to describe your samples. You have to select the tools that you want to use for the analysis. And then you press submit and you get a Jupyter notebook that is executed already for you with a report of your analysis. So this has been very successful. We also offer free RNA-seq alignment in the cloud. And so far we aligned about 50,000 samples that the community uploaded. And then this also is like a widely used resource from the group. And then a few years later, Daniel Clark joined the lab and he came up with this idea of making BioJupies even more generic and customizable. And this was this idea of appeters. So appeters are a way to take your Jupyter notebook, your workflow, your bioinformatics workflow, or any type of data analysis workflow that is coded in Python in a Jupyter notebook, and then compile it with this Appeter software development kit. And then it builds a web app for you. And then that web app allows other users that don't have any coding skills to come with their data, upload it, and then the Appeter is executed. So this is how it sort of like, what is an Appeter? You create this Jupyter notebook, but your global variables in your notebook are substituted with those meta components that are specified in this code. And then it tells the compiler of the Appeter that these should be web-based form elements, like HTML form elements that take your data in and then substitute those form elements, the field form elements, when it executes the notebook. So this builds this customized persistent report. So you come in with your data and you specify your parameters and the way you want to analyze the data, and then you click submit, and it creates this Appeter for you. And so far, we already created about 100 of those apps, and all of those are served on this Appeter's catalog that provide different types of analysis. I'm going to talk about one of them, and this is sort of like the topic of the presentation. So this is an Appeter that we are very excited about. It's called the Tumor Target Screener Appeter. So this is the splash screen of the Appeter, and these are the authors. And the idea is that you come here with your tumor data from an individual patient, and then what we do, we try to find genes that are highly expressed in the tumor and lowly expressed across normal tissues for multiple background databases. And I'm going to explain that a little bit more. So the motivation for this Appeter is from this paper. And this is a paper that was published by a team at CHOP. It's Children's Hospital in Philadelphia. And they identified this gene that is a self-surface glycoprotein, GPC2, as a potential target for immunotherapy in neuroblastoma, because when they looked at this gene in the GTEx normal tissue expression portal, they found that this gene is highly expressed on the tumor and lowly expressed across all other normal human tissues. And this is really possible. There are some genes that are only expressed in development, only expressed in extreme conditions, and they're hijacked by tumors and then used by the tumor to evade the immune system, evade cell death, and support the tumor's function. But they're also, I guess, place for you to come and attack the tumor, a weak point of the tumor where there is this surface protein that you can use what it's called ADC, and I'm going to talk about that a little bit. So ADC is an antibody drug conjugate that you can use to kill tumors by taking a toxic small molecule and conjugate it to an antibody. So first you want to create the antibody from either the cell that or the protein that you isolate from the tumor. In many cases, the antibodies already exist for some genes. If the gene is mutated in the tumor, you may need to create your own specific antibody, which is called a neoantigen. And then once you have that antibody, you can conjugate it to a drug, and then you can infuse it to the patient. And the hope is that it will find selectively those cells that express the surface receptor and then kill the cells. So this is what it looks like in drawn to scale. So the drugs is much smaller than the antibody, and then this is the linker, and the linker is attached in multiple places. And the idea is that this is a cytotoxic drug that is not specific to the target, but because you have an antibody, you don't really need to worry anymore about targeting a specific target with a small molecule. You can use the antibody to deliver the drug to the target. And already in the year 2000, there were several successful ADCs that are now in the clinic, but mostly in the last few years, more of those have been approved. And there is also a growing interest in developing those ADCs. And there are currently about 100 of those in clinical trials. But the challenge is really finding the novel targets. So many of those ADCs, they target the same surface known targets. And what we tried to do with the Apiter is to democratize this idea of finding targets for immunotherapy. So by taking data from GTEx, the same way the paper that I just showed you did, we also have data from ARCHES-4 as an alternative background. So we took all of those, the 1 million samples, and then what we do a metadata search for samples from tissues and cell types, and then we compute the average expression. And because we have so many examples or samples for each tissue and cell type, can get a pretty decent average for the expression of each gene in those cell types, and then we can use that as an atlas or background. We also have processed data from the Human Protein Atlas, Tabula Sapiens, which have single cell data. So we had to bulkify the Tabula Sapiens data, as well as other proteomics and transcriptomics backgrounds. So here is the Apiter. This is the way the form of this Apiter looks like. You come in with your specific tumor samples. You don't need any controls, but you do need to have some repeats. So you come in and you upload your file with your, typically it's like 3 to 5 samples from that specific tumor. It can also be a cluster of patients. If you don't have multiple samples from a specific patient, and then you can select whether you want to do it at the gene level or the transcript level, as well as the human or mouse. And then you can select the background, which is one of those backgrounds that I mentioned. And also you can filter for membrane proteins, secreted proteins, and also you can have like the transcripts for each of those. So what it does first, it does this alignment of or normalization of the tumor expression data with the background. So this is done using a quantile normalization, relatively standard method. As you can see, that we want to have the distribution of expression across the background and tumor aligned. And then we do differential expression analysis, which is like a one-sided analysis, trying to find those genes that are highly expressed in the tumor and lowly expressed in the normal background. And then we also apply this filter of like this membrane filter, because we are interested in targets for immunotherapy. So the results of those types of analysis look something like this. You get a table of ranked targets, and then also a box plot with whiskers about the average expression of the gene in the tumor compared to all other tissues ranked by the level of expression of the genes in that tissue. And this is just the default example. And then more recently, we created this web app that does the same thing. So here is the input form. The web app is called Target Ranger. And here you can come in with your sample expression from the cells that you want to remove. You select the background and then you submit, and then you get a rank list of potential targets. So one application that this potentially can be useful for is for removal of senescence cells. So there's a lot of interest in the fields of aging research about the potential of selectively removing senescence cells to delay aging and associated disorder. They show that if you are able to target those cells in a mouse, you get health benefits and improve health span and lifespan. So what we've done, we took data from 6 replicative senescence in vitro studies, and then we put the data into the pipeline to identify potential targets for immunotherapeutic removal of senescent cells. So this is a little bit of more of an extensive explanation of the workflow. So we use 3 backgrounds, the GTEx, Arches 4, and Tabula sapiens. We take all of those senescence, replicative senescence cells. We have 38 groups of samples from those 6 studies, and then we compare them to the normal tissue. We apply this membrane filter, and we also look at genes that are highly expressed in age populations. So senescence cells in general, they are considered rare, but once you age, they are refusing clearance and accumulate in your tissue. So they can be detected by differential expression when you compare young versus old tissues. So we also have done that with the GTEx data and identified genes that are highly expressed in older tissues. And we use that as a filter, and then we get a ranked list of transcript and genes. So this is the 6 replicative senescence studies, the cell types that were used in those studies. So those cell types were forced to towards senescence, and then these are the number of samples that we use from each study. So this is uploaded to the Apiter, and then this is the results. So you get ranked list of membrane proteins. We also have 25 top secreted proteins. And we also compare this to other publications of senescence, longevity, genes. And we found that we have significant overlap with prior publications, like there is a Sen Mayo data collections of genes, and there is this other source called Gene Age that looks at aging genes. So we get significant overlap with those studies, but we also get a lot of novel potential targets that people have not paid attention to. We also apply this pipeline to cancer, and this is the most obvious application, and specifically to the CPTAC cohort, which is not published yet. So CPTAC 3 has about 1,000 patients that were profiled with transcriptomics, proteomics, and phosphoproteomics. And by first, in order to apply it to cancer, this is like a pan-cancer. So they have, I think, 10 different cancers for those 1,000 patients. So we cluster the patients for not by only by their cancer type, but also within each cancer, there are subtypes. So we want to find those subtypes and use groups of patients in a specific cluster for target identification. And then once we have identified those subtypes, we run it through the pipeline, and then we get rank list of targets. The nice thing about it is that you can also validate the targets at the proteome level because they have proteomics data. So this is just a UMAP of the various clusters in each of those individual tumors. And this is what the results look like for applying it to the breast cancer and this kidney cancer. So this is the ranked targets. So some targets, they are showing up as in the top across all of the clusters, and some are very cluster specific, and they want to have a star in them or 2 stars in them. Those are also validated at proteomics. So the 1 star means that they are expressed at the protein level, and 2 stars means that they are specifically highly expressed in that particular tumor. So those are the best potential targets for that subtype. So it's just a little bit of limitations. I'm going to check my time. Yeah, I'm perfectly with the time. So some of the limitations of this approach, it's done at the RNA level. So when we put in the data, it's, we only can handle RNA level, but the targets are typically proteins, and we know that RNA and proteins don't always correlate. We have not done any experimental validation of the approach. And then some of those targets, like you saw, they may not be specific. They show up all the time. So we need to really watch to see why. And then the idea of creating ADCs and CAR T cells is not trivial. So this is something that we need to work with collaborators and potential pharmaceutical companies to see if that can happen. There is still potential to apply to other diseases besides cancer and aging. For example, we're thinking about removing foam cells from atherosclerosis. And then the advantage of this approach, it can really be applied to an individual patient and an individual tumor. For example, in melanoma, what we know that you get different tumors even for individual patient. So you could target individual tumor within one single patient with different ADCs or CAR T cell type of approaches. especially also across patients. So that's the advantage of this. And then it can be extended. So you can potentially identify transcription factors that are specifying the various cell types. You can identify cell lines that are most likely similar to your tumor. And there's other ideas that we have to make it better. So since I have a few more minutes, I'm going to just tell you a little bit about one more topic that I'm interested in, completely different from what we discussed. So in 2008, I attended a meeting at Googleplex, where that meeting is called SciFu. And that meeting was about bringing in different researchers from across disciplines, as well as people that are interested in science in general. And the Google founders were there, and like a bunch of famous people. And I was sort of like, I got invited there because my PhD mentor didn't really want to go. He was invited because of the paper that I showed you from Science. And then what I was taking a lot of notes. I was very quiet at that meeting. I didn't really speak because I didn't think I knew anything, but I was taking a lot of notes. And what I noticed is that across disciplines, so the meeting, the way it's structured, people just sign up and talk about their own science. And I realized a lot of those different sciences that different researchers, those top researchers, they're basically talking about the same thing. Their methods of analysis are similar, but also the system that they studied have a lot of commonalities, like in economy, in I guess it was neuroscience, molecular biology. All of those systems, they share some properties. I was interested in this concept of complex system theory. And a few years later, I was published those notes in this article called Complex Systems Biology, where I tried to compare the properties of a human cell to just general properties of complex systems. So here are the various, I guess, features or things that a cell has, and then how those can be generalized into things that you see in other engineered systems. And then, for example, there is a clock, there are sensors, there are actuators, there is a CPU, there is self-repair, there is a system to remove waste, and then there's concepts like data compression and information transfer. There is a currency, like so if you think about, like, for example, how society is constructed or a city, you can map those elements to those to a cell, sort of like a prototypical system that is a complex system. And the other thing is that I found more recently is that there is this organization of the system in layers, and the system learns, and every learning system is organized in this way, where you have like those top layers that are learning things fast, and then the only information that propagates down sort of like become more stable. So all of these ideas sort of like further expanded them and published this self-published book on Amazon. So I realized it's very easy to publish your own book on using KDP. So I created this book and published at the end of last year. So I want to advertise for it. If you're interested, I can also send you a free copy. So if you send me an email, I can mail you a copy because I get like free copies of that. So finally, I want to end by thanking the lab members. All of the work that you saw was obviously done by the members of the lab. It looks like a big lab, but this is because many of the faces on this picture are members of our summer undergraduate program. We have every summer 10-week program where we host about 10 to 12 undergraduates. And then the other thanks are to the funders. We have several NIH grants and also support from the Lyme Foundation, the Cohen Foundation. So that's it. I think I made it okay, exactly at 12, maybe not enough time for questions, but I can stay on if you like.
Well, thank you so much, Avi. What an incredible journey you've walked us through, how to become a leader in our field. And I couldn't actually help but thinking that you started as a software engineer, you put together databases, you actually started encouraging open source and build apertures, and you did not stop there, and you pursued a hot topic in drug discovery and pursued papers and automated them using bioinformatics tools, and you did not even stop there, and then you come out with this book and try to really achieve conceptual high-level understanding. I will buy your book. I will not ask for your free book to support you, for sure. But then I do have a question, but do you think that with all of the tools that's out there you're building, and then so many people working on drug discovery, there's no longer low-hanging fruit? Or do you think that with the tools and you're only touching the tip of the surface, and the major bottleneck for drug discovery is actually engaging clinical trials, and then that's how we can really see the impact of your software ideas and be implemented into the drugs?
Yeah, I can comment on that, in general, like you said, there is growing interest. There's a lot of people entering this field of computational bioinformatics, machine learning, data, all of this is becoming more and more centric. And I think there is also a lot of changes that are happening at the, medical field and clinical trials, and all of those things are transforming very fast. You can see that, for example, Amazon are now offering medical services through online. There is this ability for the patient to actually do research on their own about their disease. So that trend is going to continue. And regulatory agencies are going to struggle with keeping up with all of those changes. So there is a challenge of, but I think things will transform. It is very hard to stay competitive when you have much more competition, and there is all of those emerging efforts. But I think overall, it's going to also be a time where you're actually going to see some impact of, disease being cured. We also already saw that with COVID, how quickly the vaccines were able to be developed. And I think with cancer, it's in the next 10 years, they're going to see a lot of solutions. And other diseases like diabetes are going to be also tackled, and eventually aging. So staying competitive is one thing, but also I think the human collective mind is going to find solutions for those diseases relatively soon.
Just to check on time, we are over by 3 minutes. So if anybody needs to leave, we understand. There are a lot of people who have stuck around, and there are a couple questions in the chat. From where can we get the drug perturbation data on cell, mouse, and humans?
Yes, so this is we have this website called SIGCOMM Links. I can post it into chat, or I can share it somehow, but also you can feel free to email me. So this SIGCOMM Links has a download page where you can get it. You can also get it directly from the Broad Institute. There's a website called clue.io that offers the original data. Our data is more processed and ready for analysis. On SIGCOMM links. So I'm just pasting the URL in the chat for the person who's interested.
SIGCOMM links, okay, and it's on your website, so that's your last name lab.cloud. Okay, that's it, and we know where to find you if people want to have additional information. And also there's a chat from Dr. Cimino. Thanks so much for contributing to our seminar series. I have to run, but and I appreciate your talk. So yes, that was a great talk. I always like when it spans the whole translational science continuum like that, and definitely and very cutting edge, developing new methodologies and make it new applications. So all right, any final questions? Thank you for sticking around. And if you think of a question later on, you can still email. We know how to reach Dr. Mayan. Well, seeing none.
Can I ask you a question?
Oh, go ahead.
Can I ask you a question?
Hey, I didn't know.
Yeah, I was interested to learn how the GTX and L100 or 1000 represent the disease phenotypes that you are looking for repurposing. The GTX is not based on disease phenotype. So how's that reliable information?
So you have to come in with your profile of your disease, like a signature or some, if you, for example, if it's cancer, you take like your tumor or your blood, and then you profile that with RNA-seq or some other method like proteomics, some other method that measure the expression of many genes. And then you use those other databases for the L1000 data. It can be used to find drugs that may help, change the expression in the disease to more normal. Or the GTEx data can be used to find the genes that are specifically highly expressed in your disease that you may use as drug targets.
Yeah, thank you very much. I was interested to learn how GTX, it wasn't generated for the phenotype I was looking for, how high expression in GTX represents my phenotype. I was thinking how that's reliable information to say the gene in highly expressed in GTX is highly expressed in my phenotype.
What is GTX?
The GTX data set that is mentioned in the public domain.
Oh, GTEx. So
GTX, sorry.
Yes, so they have data from 800 patients where they measured expression in post-mortem tissues. So those are people that donated their organs for science, and then they have the expression level of their organs. So this is sort of like a normal background data that you can use to assess either the genetics or the expression of data. So it's used as a resource for the background. So if you have a gene that you know how it is expressed in your sample, you can go to their website and then you can see how that gene is expressed across all of those tissues. And that can give you some clues where that genes function. So if I understand your question correctly, but if you are, I don't know if can you rephrase your question? Is that helpful? I think you're muted, Ted.
So, sorry, that was a good answer. Thank you very much. I appreciate your work. Thank you.
No problem. Thanks a lot. And then feel free to ask me by email, like you can find me, and then I can give you a more specific, correct answer.
Yeah, thanks. Jake had to leave also. There's a few people left. Any final questions before we sign off?
I asked a question from where we can get the cells. So I would like to have some insight on that. I'm already writing to you, so I will connect you on the sidelines. Just one thing. I want to collect this data to it's a similar kind of setup or the same patient setup. So cell has been taken up from the tumors, and the mouse model is also done from the same patient, as well as the human effects are also there. Is there any kind of study like this? Because I have been trying to find it for long. So now I have started to talk to people if an industry is doing this, because this is very hard to do. And if it's not there, then it's time to write a grant like this, where we can conduct such trials.
So you have a mouse model. Is that like from a cancer? So
Yeah, from a cancer, for an example, like it can be any disease, but from a cancer. So we have a cell culture from the same cancer and the mouse model from the same cancer from the same patient, and then the drug effect on the same patient.
I see. Yeah, I think there is not a lot of work on drugs. I mean, there are some people that are trying to they take the tumor from the patient, they also put it in a mouse, and then they try multiple drugs to see which drugs are the tumor is sensitive to, and then they use that for the patient. I know that this has been done at Memorial Sloan Kettering and also at CHOPS for brain tumors for children. So they have a system where they take the tumor out, they test it against, they put it in a mouse and make a cell line from it, and then they test about 200 cancer drugs on it, and then they prescribe the drugs that actually are working in those models. So there is some evidence of whatever, not evidence, but attempts that people are doing in those two institutions that I'm aware of, but I'm sure this is sort of like a thing that should be done more.
Okay. Thank you so much. I'm already writing to you, so probably I will get in touch with you offline.
Sounds good. Thank you.
I love how your books or your paper on complex systems started out with just your observations and your notes about different people's research. So kind of, we know everything's connected. So it was kind of refreshing to see that you were able to expand on that and that it was recognized. And I hope to see more things like that.
I think it's a really fun topic to think about, the science in that way, and it's underdeveloped.
Yeah, we tend to be so specialized and siloed that maybe there isn't a space for that. But yeah, so we always go to take a step back. Well, anyway, Jake had to leave as well. So thank you all for sticking it out until the bitter end. Thanks, Dr. Mayan, for the wonderful presentation. And we hope to see you at the next Power Talk, and have a great weekend.
All right. Bye-bye.
Bye.
Bye.